Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, a new way to study cell-cell interactions from spatial transcriptomics data. I'm incredibly pleased to welcome Dr. Kyoung Jae Wan. Dr. Wan received his BS and MS degrees in electronics from Chungang University, Korea in 1996 and 1998. He received his PhD in electronics and computer science from the University of Southampton in 2006, and he is currently interested in developing computational algorithms for single-cell and spatial genomics data. A very warm welcome to you, Dr. Wan. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I would like to hand over to Dr. Wan to begin his presentation.
Thanks a lot for the introduction. I'm very happy to talk in the Technology Network meeting. And the story I would like to talk today is about cell-cell interactions. When it comes to cell-cell interactions, we have to think about the entire tissue structure and also neighboring cells, because cells communicate within the given structure and with the neighborhood. One of the ways to study cell-cell interactions is using single-cell RNA-seq data. However, somehow we made it very difficult to study cell-cell interaction because single-cell RNA-seq protocol inevitably separate cells to a single cells. We know that if cells are separated and being alone, then they behave a little bit weirdly. So we need to think about cell-cell communication in a given structure. As I said, one of the ways to study cell-cell interaction in an unbiased way is using single-cell RNA-seq data, especially using ligand-receptor co-expression. However, besides the ligand-receptor co-expression, there are many ways that cells use to communicate with each other. One of the ways could be direct contact. Besides direct contact, there are certain other ways that cells use. And the thing that I want to study these days is about cell contact. However, how can we study cell direct contact? Obviously, it's not possible to study cell direct contact using single-cell RNA-seq because they are separated. So what we talked about was to obtain a little bit differently. So by obtaining doublet. So doublet is usually regarded as errors during single-cell RNA-seq protocol because two cells erroneously meet together and we sequence it. However, if we dissolve the tissue mildly, then we may still have a tissue which is not yet separated. So we call it physically interacting cells followed by RNA-seq. So one of the ways aim is to study cell direct contact using PICseq. So this is one of the examples that I prepared. Let's say that we have a doublet or physically interacting cells of the blue cells. Then obviously, if we have the doublet of blue cells, we know that there is one blue cell sitting next to another blue cell. But if we have a transcriptome of purple cells, then we can know that a red cell is sitting next to a blue cell. The next question that we asked was, is there any genes particularly observed in a blue cell which is not observed in blue cells or red cells? So to do that, we collaborated with Josh Brinkman in Copenhagen University, and he's an expert in embryogenesis. We obtained tissue and in mouse embryo at 7.5, 8.5, and 9.5. And we dissociate cells, the tissue mildly, and obtain the cells which is not yet separated completely by using FACS sorting. And we obtain the single-cell RNA-seq and double-seq or physically interacting cells followed by RNA sequencing. So this is the UMAP for single-cell RNA-seq, just a typical single-cell RNA-seq. And basically, based on the marker genes, we label them as liver here, foregut, midgut, and hindgut. And we also labeled uroprogenitors and definitive endodome here. And this is a typical heat map for single-cell RNA-seq. So you see this is the uroprogenitor cells, and we see the uroprogenitor markers shown here. At the same time, we annotated doublets or physically interacting cells based on the marker genes. For instance, this is the doublet of the uroprogenitor and definitive endodome, and they express both the uroprogenitor markers here and the definitive endodome marker. So if we zoom in further, then we see that this is the single-cell RNA-seq for uroprogenitor, and the uroprogenitor cells express the uroprogenitor markers here. Definitive endodome cells express definitive endodome markers. Intriguingly, we find there are specific genes. There are which is LHS5, GSC, and KH2.1, which are particularly observed in the physically interacting cells of the uroprogenitor and definitive endodome, but they were not expressed in individual single-cell RNA-seq for uroprogenitor and definitive endodome cell. These physically interacting cells show the expression levels of the uroprogenitor markers and definitive endodome markers as well. To validate it, we used the GEO6. It's kind of the rough version of the spatial transcriptomics. So GEO6 basically, this is mouse embryo at 7.5. Then they dissected the embryo so that we can obtain a block of the embryo and followed by RNA-seq data. So we have the rough version of the location of the block of cells, and we know the expression levels. So we see that the uroprogenitor markers were highly expressed at the central region of the embryo, and definitive endodome markers were expressed at the outside of the embryo. Interestingly, the niche space markers, or the genes which is particularly observed in the PICseq, was expressed at the region marked by, in between the region marked by uroprogenitor and definitive endodome, as shown here. So, based on that, we are kind of motivated to label cells based on their label cell based on their neighboring cell information. So we collected the niche-specific genes and built on surface vector machine training model to predict the neighboring cell label. So that these are all uroprogenitors, and we found that uroprogenitors sitting next to definitive endodome express genes such as LHS5 and NKH2.1, and uroprogenitors sitting next to notochord express PRTG, and uroprogenitors sitting surrounded by other uroprogenitors do not show the expression of the PRTG or NKH2.1. And we also showed that GSC shown here is expressed in definitive endodome, not in uroprogenitors. So we also tried to validate it using staining analysis. So bottom part is definitive endodome marked with FOXA2, and upper part is uroprogenitor marked with SARS2. And we found that NKH2.1, which has been predicted to be niche specific, is expressed in the uroprogenitor here, and when they are very close to definitive endodome. So we performed another validation. So we obtained definitive endodome cells and uroprogenitors, and the definitive endodome cells we marked with GFP, and we co-cultured them. After they are co-cultured, we separated them each other, and we performed RT-qPCR to see the expression levels. And we found that genes such as NK2.1 shown here is only expressed in co-culture system, and then as we predicted, NK2.1 is only observed in the neuroprogenitor cells, not in the endothelin cells. So validating our prediction. So we also wondered how long does the niche-specific genes express after they are separated into single cells. So we started to measure the expression level of NK2.1 in neuroprogenitor cells after they are separated from the endodome, and we found that after 6 hours, the level of NK2.1 has been reduced significantly. So this is a cartoon that summarizes our research. So in a tissue, we see many types of tissue, and there are definite endodome cells on the left and neuroprogenitors on the right. However, definite endodome cells are not same in that the definite endodome that are sitting next to neuroprogenitor cells express specific genes such as GSC. And also neuroprogenitors sitting next to definite endodome express NK2.1. So we wanted to further extend our knowledge obtained in PICseq or PICseq into spatial biology. And there are mainly two types of spatial biology technology. One is image-based technology, and the other is sequencing-based technology. Especially for sequencing-based spatial transcriptomics technology, we need a barcode that tells the location of cells. And because of it does not mean that a spot in spatial transcriptomics, a spot does not always match with the cell, and the performance depends on the resolution of the barcode sitting in the slide. And this slide shows the development of the spatial biology technology, especially for NGS data. And when he started this, when the technology was shown, it was the resolution was 100 micrometer, and we have the SlideSeq data in public, which is 10 micrometer resolution. And there are other technology which can measure the spatial transcriptomics data, which is lower than 1 micrometer resolution. So one of the naive way to analyze the spatial transcriptomics data is just regarding it as like a single cell analysis. So after clustering, we can annotate them and mapping back to the assay. And this is one of the examples. However, we think that spatial transcriptomics can be regarded as an image. So we try to apply image processing technology. So these are transcriptome from spatial transcriptomics data, and we perform dimension reduction algorithm by running PCA. Then we have the loading value along PC1 and PC2, PC3. The loading value is normalized to red value from 0 to 1, so that after this normalization and color coding, we have this map. And this map shows that we have the location of cell. At the same time, the color here, which is a combination of RGB, can represent 20,000 genes in a cell or spot. So after performing some other algorithms such as tessellation and rasterization, we have the matrix form of the data. Then we can perform some tissue architecture detection by applying simple KNN approach. So this is one of the results of the Vesalius, which can perform tissue architecture detection, and Vesalius successfully identify regions such as dentate stylus, corpus callosum, and inner CA field. And compared to that, the algorithms which has been developed for visual data, which has been as a lower resolution data, is they cannot perform very well because they try to find the neighboring cells and to see if the neighboring spots have the similar transcriptomic information or not. And they are slow, as you see here. The cellulose is very fast and performance is very bad. And this is one of the results. And the cellulose to identify CA123 region successfully. And also, because this is image-based approach, we try to apply some image operator. In this case, erosion, which is make it thinner, dilation, which make it bigger. And after erosion, we perform some statistical tests and we identify some genes which is morphologically defined in the tissue architecture. One of the examples is KIF5A in corpus callosum, which we predicted to be highly expressed at the central region of corpus callosum. Interestingly, we also found that in Alhambra atlas that KIF5A is highly expressed at the central region of the corpus callosum. And this is another example in mouse embryo. And left on the right side, this is eye region. And if you also further zoom in the eye region, you find some of the region highly marked with CRY-E4A at the central region of it, and PMEL, which is highly expressed at the boundary of the eye region. And this is the values applied to other spatial transcriptomics technology called the PICseq. And left side is H&E staining image. Right side is the tissue territory that we predicted. And the territory we predicted was matching very well with the H&E staining image. And this is another example. Left side is the original data in the paper. This is sick-dish data in mouse embryo. And they tried to label the tissue using their own way. Right side is the tissue label detected by the cellulose, and the tissue boundary look very clean and nice. It's not only the transcriptomic data, there are spatial cut and tag data. We downloaded that. This is the case of the cut and tag data for HDK for trimethylation. And left side is from the original paper. They performed KNN, and the tissue territory is kind of scattered. It's not real. And on the right side, you see the prediction result of the cellulose. You see nice territory detected by the cellulose when it uses the spatial cut and tag for HDK4 trimethylation data. So we try to see also the region-specific genes. So these are some of the examples. On the left side, you see all astrocytes colored in thalamus area and cortex area. And we found that they express specific genes depending on where they are located. So astrocytes express MVP when they are sitting in thalamus, but not in cortex area. And we also found interneurons express GAD1 when they are sitting in cortex area, but not in thalamus. So this is the version 2. So basically, the cellulose version number 1 used the 3-dimensional data after they are converting into RGB. So we try to use more dimensional information. So after we use more dimensional information, we could predict more, defined tissue architecture, as you see here, for mouse hippocampus and mouse embryo. So in the beginning of the talk, we talked about the cell-cell interaction. Can we study cell-cell interactions, especially cell contact, using spatial transcriptomics data? Yes, so we developed a tool called CellNeighborEX, and CellNeighborEX used different strategy for image-based data and NGS-based data. Excuse me. For image-based data, CellNeighborEX try to find a cell which is surrounded by the same cell type. At the same time, we try to find a cell which is surrounded by at least one different cell type. And we perform the statistical test using the heterotypic neighbors as a foreground and homotypic neighbors as a background. And this is one of the examples. This is a sick fish data in mouse embryo. And we labeled the gut tube cells using color cyan color. And these are gut tube cells. And these are also gut tube cells. However, they are different because their neighbors are different. So these are gut tube cells next to cranial mesodome, gut tube cells next to neural crest, gut tube cells next to other mesodome cells. And we observed that gut tube cells next to cranial mesodome express TBX1, gut tube cells next to neural crest express PICx1, gut tube cell next to spatial mesodome express FOXF1. So we try to visualize it by showing gut tube cells next to cranial mesodome with a red circle and gut tube cells next to neural crest with green circle, and gut tube cells with spatial mesodome with the blue circle. And we colored the inside of the circle using the combination of TBX1, PICx1, and FOXF1. So if we see the red circle with inner red color or green circle with green red color, that means that our prediction is correct. So as you see in this figure, red circle is always with the red color inside the circle, and blue circle is always the blue color inside the circle, visualizing our research. And there are certain type of there are NGS-based spatial transcriptomics data, as I explained before. So SLIDESeq is about 10 micrometer resolution, which is round cellular resolution. However, we cannot tell that one spot means a cell. So we try to use the kind of the concept that we use for physically interacting cells, or doublet. For instance, let's say that left side is a red tissue, right side is a white tissue, and a spot from red tissue will have the red color, red color transcriptome. But at the center, we will have the transcriptome, which is a mixture of the red transcriptome and blue transcriptome. And we try to use that as a surrogate of the cell-cell contact. So we try to see the specific expression at the heterotypic beads, which is not expressed in the homotypic beads of the A cells and B cells. Then we can identify cell contact specific genes. The next question is, then how can we know that the cell specific genes is from A or B? So we actively use the heterotypic beads in this case. So BTI1A is one of the genes that we identified to be specific when monocyte and hepatocyte form on heterotypic bead. So we try to show the expression levels of the BTI1A, and on the x-axis is the proportion of the monocyte and hepatocyte for the bead. So when we investigated that, we found that whenever there are more transcriptome for hepatocytes, the BTI1 level decreases. It means that BTI1A is expressed in the monocyte when they are very close to hepatocyte. And this is one of the examples in mouse brain. So we observe that DDI1, TRF, and FABT7 are highly expressed for the heterotypic beads for endothelial tip and astrocyte. And we also predicted that FABT7 is expressed in astrocyte, but not in endothelial tip using the regression analysis. And figure B is showing the co-culture result and also the test that in co-culture, and after they are separated, FABP7 is only expressed in astrocyte, but not in endothelial cell. And figure C is the visualization again. So in this case, we show the heterotypic of the endothelial tip and astrocyte with a red circle, and FABP7 is particularly observed in the heterotypic phase here, which is shown in the red circle. This is another example when we apply CellNeighborEX to liver cancer data in mouse. We found two cancer regions marked by R1 and R2. And we found that the gene F13A1 is particularly observed when they form heterotypic pairs with monocyte and tumor cells. And when we visualize that, we are interested to find that the heterotypic pairs for the tumor cell and monocyte were located here. And F13A1 is highly expressed only for the heterotypic pairs for monocyte and tumor cells. And this is another example for in mouse brain. We also investigated the heterotypic pairs for endogenous cells and other cell type, and found that endogenous cells expressed NAV when it forms the heterotypic pair with choroid, and endogenous cells express GDA whenever it is next to neuron. Endogenous cells express ATP 2B1 when it is next to each neuron. And we validated that using single cell RNA-seq data as well, because, well, these cells like NNAT and ATP2B1 is expressed only in endogenous cells when we look at the single cell RNA-seq data. And this is also validated using the regression analysis as well. So one of the wide way to study cell-cell communication is using ligand-receptor expression. So we try to compare our result with the ligand-receptor-based predictor. One of them is called NISHNet. And NISHNet, we use NISHNet because it detect the ligand-receptor co-expression as well as the downstream genes. So when we compare the list of the genes that we obtained using our approach, which is CellNeighborEX, and the result of the NISHNet, we found only a little overlap. So there are overlap. However, the lists are quite different. It has that the cell contact mediated communication is an independent pathway, which is quite different from ligand receptor mediated communication. And at the same time, we are trying to integrate several spatial transcriptomics data. For instance, so we have the reference image data that's shown here, and we have the query data shown here. Can we integrate it together? So we try to integrate them using the centrally located cells as well as the neighboring cells together, which is quite different from other data in that we use the neighboring cell information more actively compared to other algorithms published in they are in bioarchive, but with other data. So basically, so we try to use the transcriptome of the centrally located cells and neighboring cells. When we show the aligned cell, we see that this is the aligned information of the query set to the reference set, and we see that, well, these are well conformed. And also we try to integrate the DNA-seq data and RNA-seq data together, and it worked really well. So another question that we wondered was, can we predict the location of the single cells by reading only their transcriptomic information? So basically, we use the deep learning approach as shown here. And this deep learning approach is trained using the spatial transcriptomics data, which could be SLICIC, which is high resolution spatial transcriptomics data, or it could be Visium, which is low resolution spatial transcriptomics data. Especially for Visium, we need to increase its resolution, and we thought that this is one of the ways to enhance the resolution of the Visium data by integrating single cell RNA-seq data. So this is the cross-validation. You see the reference. For this cross-validation, we use the 80% of cells for Bs for training and 20% of the Bs for testing. This is the result of the 20% of the Bs. So when we trained using slice data in mouse embryo, we found that in SC2-SPA, which is we developed for using deep learning-based approach, could predict the location information roughly for this cross-validation, but when it applied other algorithms such as Ngram, NovaSPA, or SPA OC SC, and this is a regression analysis, they did not perform well in predicting the location of Bs in this case. So this is the example. So we trained the neural network using Visium data and try to predict the location of the single cells, which is obtained from the same mouse and also from different mice. And right side is the example when we show the prediction result, which is using the single cell RNA-seq data from different mouse brain. And you see here, so spatial transcriptomics, this is Visium data, show the expression levels of RGS9 shown here. And when we visualize the prediction result, it says that RGS9 is shown here. So it basically matches well with the spatial transcriptomics data. So as a conclusion, so we think that cell contact is an important way for cell communication, and we actively study cell contact using doublets and also using by developing CellNeighborEX. And we believe that neighbor information is encoded in the cell transcriptome, as also the global information is encoded in the cell transcriptomes. So that's why we could develop an algorithm of SC2-SPA, which basically map single cell RNA-seq data to the location in the spatial assay. And for this work, I would like to thank Patrick Martin and Kyoung Kim, who developed the Vesalius algorithm and CellNeighborEX algorithm, and also Limbo Liao, who developed SC2-SPA, and Junior Kim, who developed the algorithm to detect neighbor cell information using physical interactions. And I would like to thank all my funding agencies and my collaborators. And thanks a lot. And happy to take questions.
Thank you very much, Dr. Wan, for your incredibly insightful talk. As with all the talks today, we are likely going to have lots of questions from the audience. I can see that we have received some questions already, so I'll move straight on to the Q&A section now. So, Dr. Wan, your first question is, what is the biological mechanism involved in neighboring cell-dependent gene expression change?
Yeah, besides ligand-receptor co-expression, there could be other ways that cells could use, such as gap junction, and ligand-receptor co-expression is also used for the neighboring cells. And also cells use, there could be some other ways cells detect, which we do not know clearly. For instance, whenever a cell is physically pressed, field pressure, then they usually show different gene expression. So there are other ways that cells detect the physical environment as well as with the neighboring cells. So there are other ways that we can think about when it comes to cell interaction.
Thank you so much, Dr. Wan. An excellent answer. And we have time for a few more questions today. So moving on, your next question is, can CellNeighborEX be applied to all types of spatial transcriptomics data?
So basically, CellNeighborEX can be applied to image-based technologies such as Morphish or SIGFISH, and it can be applied to cosmics data. And for the NGS-based data, it can be applied to slide-seq data or stereo-seq data, which is higher resolution. However, it cannot be directly applied to Visium data because Visium data, its resolution is about 55 micrometer, and it can contain more than 10 cells inside. So it's a little bit hard to apply CellNeighborEX to Visium data.
That's excellent. Thank you, Kyoung. And we have time for one final question today, which is, how is the spatial transcriptomics data integration applied to cancer research?
Basically, we can think about certain scenarios. So we try to treat cancer cells using a lot of chemotherapy or immunotherapy, but still we found some responders and non-responders. And one way to define responders and non-responders is to use the mutation information. However, there are certain cases where we cannot directly use the mutation information. However, if we have this spatial transcriptomics data, for instance, we can see that in certain cases, we see that drug works, so responders and non-responders, based on their environment. So if we integrate spatial transcriptomics data, then if we have data before and after treatment, we can study the microenvironment of cancer cells to define, for instance, responders and non-responders.
Thank you so much, Kyoung. Unfortunately, that's all that we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thank you once again, Dr. Kyoung Jae Wan, and we'll be back soon with the next talk.